Hopefully we have enough space on in the cloud or whatever. But anyway, so I'm going to talk to you about the Mexican bean beetle and some viruses. So one of the things about insects is that, you know, plants are responding to chewing insects, right? And piercing, sucking insects. You all remember me talking about this? Well, it's not just the damage that the insects has caused. We know that saliva is a factor too, right? And what else is important is the fact that when you wound yourself or you wound a plant, those wounds can be holes that allow for pathogens to seep in. Like if you cut yourself, one of the things you're concerned about, if you damage your skin, right, is that you might get infected. Well, there are some insects, not only is there that damage that can get in there accidentally through the environment, but insects can also transmit diseases to plants. You know that insects can transmit diseases to you, right? Mosquitoes, well known for spreading things like malaria, St. Louis encephalitis, Western equine, and I don't know all the different ones, equine encephalitis, but all sorts of different diseases. Ticks can spread different diseases as well. So plants have to deal with two pest problems if you have an insect that spreads a disease. Does that make sense, everybody? So that's the thing, is that you have to deal with two potential pest problems. And I'll get up in a minute, but here we have Mexican bean beetle. Epilancha vervestis is its scientific name. And what does it look like to you? Yeah, it looks like a ladybug. So it's part of the same family, Coccinellidae. However, most ladybug insects are often predators, so they'll feed on aphids. So a lot of times ladybugs are actually considered a good thing in agriculture. However, not all of those ladybug types in the family Coccinellidae are actually predators. Some are herbivores. And so herbivory is caused by this beetle here, the Mexican bean beetle. And what do you all think it might feed on? Beans, very good. Yeah, they feed on beans. And they do so in an interesting manner. They, when they're eating on beans, if you look at this leaf here, you'll see that it kind of leaves a lace-like appearance. You see there's still, like the caterpillars will chew the leaf off at the end or make a big hole, but you can see that it's kind of got the vein still. So it leaves kind of like a little net-like appearance. And so what they do is they scrape the surface of the leaf and then chew it. So they still have chewing mouth parts, but instead they scrape the leaf and then basically drink the juice. And so they're a little different. The interesting thing about lady beetles as well as the Mexican bean beetle, you know, because again, they're in the same family, but one's a herbivore, one is, and there might be, I don't know how many species of lady beetles there are, but this particular one is a herbivore, you know, exclusively, is they can kind of protect themselves from predators that try to eat them, because they can emit kind of a noxious taste and a kind of a leak blood through their appendages. So that's kind of what that orange stuff is an example. So it's like a self-defense, so that when they get attacked, their joints will leak this nasty flavored blood, whatever chemical they happen to have in it. By the way, the name of blood in insects is called hemolymph, if you didn't know that. So they don't have a lot of blood cells like we do in our blood. Insects are mostly water, and they do have blood cells that might help in defense. But remember, our blood cells have proteins called hemoglobin in it that travel through veins and arteries. Well, insects have what you call an open circulatory system. And so that means they don't have veins and capillaries and arteries for the most part. And the blood is just kind of circulating in the body cavity. So that means to get oxygen, they use those little, remember those, the spiracles with the trachea and the little tracheoles, the little tubes that go through the body. That's how they get their oxygen. So they don't need red blood cells with hemoglobin like we do. Because remember, hemoglobin is what transports the oxygen in ours. So anyway, just kind of give you a little heads up on how that they do that. Now, is there such thing as a baby beetle? There isn't a small beetle, they're larvae. So here we have a Mexican bean beetle larvae. And this is the lace-like damage that's caused to leaves. See there's lots of this is basically they're crushing it and drinking the juice. They're not really tearing off the whole leaf part, so the veins are still there. So quite a bit different type of damage than what you would see from a caterpillar, for instance. So it's more crushing and drinking and so forth. And so here we have a Mexican bean beetle larvae on the left. That would be one that's probably really close to molting and becoming becoming a pupa and becoming an adult. There's no such thing as a baby beetle to become a bigger beetle. Once they've reached the beetle stage, they're an adult. So they'll start off, they have 4 instars. Remember, each instar is a molt. So they're gonna have an egg. Then you're gonna have your first larval instar, second, third, and fourth, and then it becomes an adult, become a pupa. And this usually takes place over like 2 or 3 week period. Remember, insects are ectothermic. That means that they grow based on the temperature. So you'd call them cold-blooded, be another way to call it. But so that means they'll grow faster in the hot July weather than they will grow in maybe in May, for instance. It'll be much slower. So they'll have much more, so they'll have babies. There's more food then too. And of course, that is growing at a much faster rate. So it comes down to being a serious pest by just having the fact that they can lay thousands of eggs. But how do the beetles know what plants to lay their eggs on? Again, it's going to be based on a lot of things like the smell of the leaves, the taste of the leaves, the UV light, the visible light, all these kind of things probably influence where the mother will lay her eggs. Now you've all seen this before. What is this again? These are our plant defense pathways, and these are put in a very simplified version. There's actually lots of chemicals involved in this pathway, this biochemical pathway. It doesn't just go straight from system to jasmate and straight from jasmate to plant defenses. Remember, there's lots of different chemical steps that can turn on a variety of chemical defenses. There might be hundreds of chemical defenses that are formed and numerous steps before it gets to this. The induced resistance pathway, or also known as the IR pathway, is the defense pathway that's primarily geared towards wounding of the plant and chewing by herbivores. And then some pathogens can stimulate it, like some fungal pathogens may trigger this pathway, or bacteria may trigger this pathway. But the majority of pathogens tend to trigger more of the SAR pathway, or the system-acquired resistance pathway. The systemic signal is hydrogen peroxide that forms, again, it'd be a complex chemical pathway, but ultimately can form salicylic acid. You remember, this is either can also be called salicylate. Jasmate can also be called jasmonic acid. But anyway, I'll go through a variety of chemical steps and then ultimately become plant herbivore defenses, or excuse me, plant pathogen defenses. Again, it's primarily geared towards dealing with viruses. It's primarily geared towards dealing with viruses but it can also deal with a lot of bacteria, and it can deal with some insects. The insects that tend to stimulate this pathway are what? Do you remember? Aphids and whiteflies are good examples. Now, aphids and whiteflies are very small, delicate insects. They have mouth parts that are like straws. So they're kind of like the mosquitoes of the plant world. You know, mosquitoes are pretty small bodied, kind of weak, but they have that strong mouth part that can suck your blood. Well, aphids are small little insects that drink the sap from plants, or the nectar from plants, whatever you want to call it. And so is whiteflies. It's kind of the same thing. It's a delicate little insect. The interesting thing is they're drinking so much sap that the sap's constantly going through their body, that some ants actually guard them. Did you know that? Some ants guard aphids, and then they'll drink the waste products of the aphid while they're basically protecting them. And so the other thing though is this sap is, they have lots and lots of aphids. There's a lot of sap, and that sap can actually get mildewy and cause infections to plants also. But the thing about aphids and whiteflies is they can spread viruses. Hegemony viruses, I believe, are spread by whiteflies. So that means that in the case of aphids and whiteflies, this plant virus will actually infect the aphid or the whitefly, so that it actually becomes infected inside the aphid or the whitefly. Then, when the virus builds up inside the aphid or the whitefly, they'll suck one of these plants and deposit the virus in the plant, and the plant becomes infected. So it makes you kind of think a little bit. Why do plants typically turn on these plant pathogen defenses against aphids and whiteflies? I think it's in part due to protecting themselves from the viruses that these aphids and whiteflies are probably spreading. So it's kind of like dealing with two problems, right? They got the aphid feeding on it, but the even bigger problem when aphids are around is they're spreading viruses. Well, let's get back to the Mexican bean beetle. The Mexican bean beetle also spreads viruses, but it does it a little differently. The Mexican bean beetle does not become infected by the virus. Instead, the virus sticks to the mouth parts, the mandibles, where they're chewing insects. They'll stick to them. There's a couple different types of viruses that come with this beetle. Southern bean mosaic virus is one, and the other one is bean pod monovirus. So those are the two that we'll see in a second in the next picture. But this makes me kind of wonder a little bit. Again, this is research that I did. What pathway do you think this beetle is stimulating? Naturally, I would think that the pathway would be primarily the induced resistance pathway, but the beetle is also spreading a virus. And here's another question I have for you. Is it beneficial for insects to spread viruses to plants? So one person in the back said no. Again, this is just your hypothesis, right? You don't know for sure. There could be pros and cons. One con is if you're an insect spreading a virus to a plant, the plant is your host for both the virus and the insect. And so now that you're competing over the same food source. So that's probably one of the reasons why you might say no. But if it was so bad for the aphids, you'd think they would avoid the infected plants and try to go to another plant, and maybe they do. But there is some evidence that suggests that virus-infected plants can be nutritious to a certain degree, depending upon the amount of infection. And so the question is, can it be beneficial to infect a plant? Because if you infect the plant, let's say if you're the beetle and you infect the plant and you turn on this pathway of defenses, maybe you can avoid some of the more detrimental defenses over here. Because remember, bean plants might stimulate more trypsin inhibitors that block the beetle's ability to digest proteins. So if the beetle spreads this virus, could it turn on this pathway and maybe get a little bit more food or nutrition out of the plant because it's no longer turning on these defenses? Because remember this crosstalk, do you guys remember that? So if the virus is spread by the beetle, maybe it's turning on this pathway and inhibiting this pathway. So maybe that's a benefit. We don't know that for fact. These are just hypotheses and questions that you can ask as a researcher. But there might be other reasons. Maybe infected plants are less nutritious. Maybe they take up all the proteins to make more viruses, and so that's not nutritious to the plant. That might be a counter to that argument. See, there's lots of potential possibilities, right? And again, it depends how infected the plant is. If the plant's about to die, it probably isn't a very good food source. But if maybe it's just a little bit infected, it's a good food source. This kind of depends a little bit. So these are the kind of questions I asked with a plant virologist over at University of Arkansas named Dr. Rose Gergerich. And so again, so we call this a tritrophic interaction. I don't know if you ever heard of this or heard of anything called a tritrophic interaction. Tri, of course, means three. So we have the beetles, that's the group of the insects. We have viruses, which you could argue isn't an organism technically, right? And we have plants, which are the hosts. You know, it's technically, a virus technically is not alive or not a living organism, but it's very organismal-like, isn't it? You can replicate and so forth. But the plant has to be wounded, it has to be damaged for it to enter into the wound before it can start infecting the plant. So again, here are our three species. Well, again, the virus isn't. And so let me tell you about a little bit about some of the bean. We worked with the black Valentine bean and the pinto bean. And here is our beetle. You can see the lace-like damage. You can see the eggs. You can see there'll be 4 instars for the larvae. Then we have this become pre-pupa. This is actually a pupa. And then it goes through what we call complete metamorphosis into a beetle. So I'm going to ask the question is, what happens when this beetle transmits this virus to these plants? Is it beneficial to the beetle? Is it beneficial to the virus? It's obviously going to be a little bit of both. But the main thing is that these insects do not become infected like aphids do. It just sticks to their mouth parts. But every time they eat, it comes off their mouth parts. So they have to eat an infected plant for it to stick on their mouth parts. This is what the infection looks like. For bean plants, the leaves become mottled and damaged. This is what we call mottling. So what you see here is lack of chlorophyll. So you see dark green leaves here and mottled colored leaves there. So it's clearly going to, without that, the green stuff is chlorophyll. Without the chlorophyll, the plants become stunted in their growth. Now let's talk a little bit about viruses in plants. A young plant that's infected is usually in trouble because it gets stunted in its growth, it doesn't grow properly, and then the bean pods are all mottled and don't look healthy and you don't eat them. There's nothing, it's not worth harvesting hardly. Now the flip side is if the plant's mature and pretty grown up, usually the virus really doesn't use, at least in this particular virus, doesn't really take off in it. So you might get a little bit of infection, but for the most part, plant can withstand it and can move on and still have a pretty reasonable harvest for the growers. Does that make sense, everybody? Here's something kind of interesting to you. It might be interesting to you. You can take let's say, with like vaccines, you could take a closely related virus that isn't exactly this virus, for instance, and you can scrape a plant and add it to give it a little minor infection, and it'll make it more resistant to other viruses that come along. So you can kind of vaccinate your plants. Is that kind of interesting? That's good. Okay, so here's our objective. We are interested in determining whether a virus and its beetle vector benefit each other by co-suppressing plant resistance. Just to make sure, back up, just make sure you understand what I'm talking about. Remember the two pathways? Remember how there's crosstalk? So if the virus stimulates the pathway, maybe it suppresses the jasmonate pathway. If the beetle feeds, it maybe suppresses and so maybe that's beneficial. So the beetle comes along and is eating the plant, really stimulating jasmonate levels. Maybe the plant becomes more susceptible to a virus later. That's just a question. You can make another hypothesis that it isn't. And that was the hypothesis I made. Or if the plant becomes infected, maybe it becomes a better food source for the beetle because the plant defenses are being suppressed. That's one possibility. Maybe it isn't. Maybe the viruses, destroy the plant and it's not very edible. But these were the kind of questions I asked with our virologist. Since I did plant herbivore interactions, she was interested in plant virology, but this was what she worked with. So that's how I got on board and pulled the idea to her. And I took a plant virology class. So anyway, for short, bean pod model virus is known as BPMV, and southern bean mosaic virus is known as SBMV. So those are the two. And so again, it's important to write hypothesis statements that you can test. The less complicated your hypothesis is, the more clear cut it is, and your testing makes a better experiment. So that, and I'm hoping to make you think about experimental design a little bit in this class. And so here's my hypothesis that I put together. The Mexican bean beetle larvae will have a greater body weight upon feeding on virus-infected plants than non-infected plants. Very straightforward hypothesis. My hypothesis, based on what I think might happen, is that the virus-infected plant will be a better food source for my beetles, at least for a reasonably healthy plant. And so everything being equal, but one's infected, one isn't. My hypothesis is that beetles will grow better on the infected plant, grow faster. The null hypothesis is what you think won't, will be the opposite essentially. Or let me back up. It's basically stating that nothing's going to happen because of your treatments. Okay. So the null hypothesis is there's no difference whether the beetle feeds on a virus infected plant or on an infected plant. Does that make sense? Non-infected or infected. There's no difference. That's what we call a null hypothesis. So the Mexican bean beetle larvae will have equal body weights regardless if they feed on the virus infected plants or the non-infected plants. The alternative hypothesis is if we see something opposite of what the hypothesis is. So the other possibility is that the beetles will grow faster on healthy or non-infected plants than infected plants. Does that make sense, everybody? So I'm going to test this hypothesis, but these are possible conclusions that will happen or could happen. Does that make sense? I can't think of any other type of hypothesis you would have, but these are. And again, this in science and particularly experiments, keeping it simple, the KISS principle, keep it simple, stupid, is the best way to develop your hypothesis. So we had to take care of growing the beetles in a greenhouse. This student here is letting them feed on pinto beans. And the reason why we used pinto beans was initially, before we used the black Valentine bean, is that these plants don't get an infection from southern bean mosaic virus or bean pod monovirus. These plants stay essentially, they might get a little bit of localized infection, but it doesn't spread through the whole plant systemically. So that means we can keep the beetles relatively virus free. Does that make sense? So that's why we're going on pinto beans initially. And this was one of those students that you remember from the big group. He stuck around and did some research with me later. And now he's, probably hitting close to 40, late 30s anyway. He was a pothead at this time that he actually got out of that and has a family and everything. So there's still hope for some of you, I know. I was amazed that and again, I'm just saying that your life isn't over at 19 or 20 or just starting, so. I thought it was really cool to see that. Okay, and then here is one of my former students who helped me a lot, Spencer Williams. And what they're doing here is they're inoculating plants. These are black Valentine beans. And so what they did was they took a substance called carborundum. It's kind of like a little silvery look appearance. It's like a little gritty sandpaper. It's actually made from the diatoms of, diatoms are found in oceans. They have silica, and so that silica is a little bit sharp. So they'll take a little bit of that, silica, diatoms, whatever, carborundum, rub it on the leaf, and it causes little scrapes on the surface of the leaf. Then they can take sap that's infected and rub it on the leaf and basically make an infected plant without the beetle. We can take some sap from a plant that's infected with bean pod monovirus and rub it on the leaf and cause that plant to become infected. So here they are shaking it on and rubbing it. Now we have an abundance of infected plants. And so what do you think would be a good idea? We should have plants that aren't infected as well, right? But we'll still rub carborundum on them just because that might affect the plant defenses or something. When he was a high school student, he actually took one of these projects with him to International Science Fair in the world. I'm not sure what he's up to lately. Anyway, so this is a growth chart. So what we did is we took these infected plants, we put eggs onto them, we let the larvae grow, and then we weighed them. Does that make sense, everybody? And then we got their mean weights. We took measurements at day 7 and day 14 of the beetles. By day 14, they're pretty big, like 4th instar before they come a pupa. So what you're going to see here is a line chart. So we weigh the same beetles over time. And this is the mean larval body weights in milligrams. And so on day 1, they just basically hatched out the egg. They weigh the same amount of weight. Day 7, we start to see a little separation. The healthy plants are not infected. The red, hopefully you can see the red, is bean pod monovirus, and the green is southern bean mosaic virus. What do you see in this graph between these 3 dots? The body weights appear to be slightly less for the healthy plants, for those beetles, than in the affected plants. The beetles seem to be growing a little better, but at day 7, there isn't that big of a separation, is there? But we just kept watching it, and the beetles that were feeding on the virus infected plants grew substantially faster than when they fed on the healthy plants. Now, again, we're looking at probably what, 25 milligrams versus around almost 35 milligrams. So what is that, around what, 20% or something? I have to do the math. You will do 25 divided by, it's like 35. But anyway, we're looking at a pretty big difference. Now keep in mind, what's the benefit of growing faster? Well, remember when I was telling you about summers? Insects that grow faster can lay more eggs sooner and have a bigger population sooner. Bigger mothers can lay more eggs than smaller mothers. All that can make a big difference over the course of a summer. So it does seem in this experiment that it is a benefit to feed on infected plants than non-infected plants. Again, these are infected, but they still are there, right? They're still kind of bushy, they're not dying plants. So again, now the question that I have to ask you is it because the plant defenses were stunted? Possibly. Because if you can't photosynthesize, you can't produce as many plant defenses, or maybe that plant pathway was suppressed by the virus. The jasmine pathway was suppressed by the virus. There's a lot of different possibilities. Or maybe there's other things that are different. Maybe they're make the plant more nutritious. We don't really know. Because we have that hypothesis, we have that speculation. But again, then you have to make another series of experiments trying to figure out what that might be. And again, remember, this is still kind of relatively artificial. We can detect this difference in a lab, and then we can speculate that this is happening in the world, but we don't know 100%, right? Because this experiment was done in a greenhouse. So what does it really mean to agriculture is kind of hard to tell. We have some pretty good laboratory evidence for it. And it seems reasonable that I would speculate it does happen in the field at some level. But again, how many plants are infected next to it, there's a lot of different things going on. Here's a question I have for you. If the beetle is growing faster on infected plants, do you think that they would preferentially choose infected plants over non-infected plants? It'd be pretty smart if they chose the infected plants, I would think. Because that means that if your mother laying your eggs, and if mother really knows best, she's gonna want to lay her eggs on the plant that lets her babies grow the fastest. So I would make the hypothesis that adult beetles and larvae will prefer to feed on virus infected plants in comparison to non-infected plants. That's my thought. So what would be the null hypothesis? No preference. The alternative hypothesis, they prefer to feed on the healthy plants. So it's the same thing. But now I ask you all, how do you test that hypothesis? If I'm going to ask you to do this research, I want you to tell me how you're going to design an experiment to test this. And so how would you do it?
We have to make the infected plant.
Yeah, so you have infected plants.
Yeah. And keep the beetles on them.
And then what?
Make beetles on them.
So can you elaborate? So you have beetles that you got infected plants, you put beetles on them. Is that all there is to it?
Yeah, we have to make first infected plants.
Okay, so we got, yeah, so definitely you got some methodology, like you have to grow the beetle, you have to grow the plants, right? So there's all sorts of things. But what's the basic experimental design? And everybody can help on that. How would you do that? You're just gonna have infected plants and non-infected plants?
We have to infect the plant with the beetles.
Yeah.
Fresh one.
But how are you gonna design your experiment? Are you just gonna throw some plants in a room and let the beetles run around? Yeah.
Can you repeat your question?
What's that?
Can you repeat your question?
I want you to, maybe you all think about how you're gonna make this experiment. Can you think about how would you go about making an experiment that allows you to test whether or not an insect prefers a virus infected plant versus a non-infected plant? Yeah.
So what I will do in this case is I'm going to inject, I'm going to have like 3 plants, like 2 plants. One will be infected with virus, the other one will not be infected with virus, and put them separately. Then I will allow beetle with infected that has virus on it to feed on the infected one. Then the one that doesn't have that is not infected with virus, that is a leaf. We allow the beetle to grow to feed on it. Then I'm going to start monitoring the weight. If it grows on the infected one, it means this is preferable. If it doesn't grow on the other one, it means I'm going to sideline it, which is the alternative hypothesis that I'm going to go with my lead hypothesis.
Does anybody want to add to his idea? Yeah.
You can also compare if the beetle likes it by monitoring how the leaves look, right?
Mhm.
So if it's not that much like lace look on the leaves
Yeah.
of the infected or the uninfected, then it means that.
Yeah.
it's the reverse, which one.
Yeah, that's another good idea. Yeah. Yeah.
Do you have like the plants separate or together to see like the preference?
Yeah, so that's a good question. I think that if you're just trying to look at a choice, they should be relatively close in proximity. You know, his point was that maybe some beetles are infected versus some that aren't, which would be another kind of layer to add to it. But remember, the virus, they don't stay infected, they just put it on their mouth parts temporarily, but it might affect their behavior, I don't know. But having them in close proximity and then letting the beetle kind of wander around and feeding on one plant or another and seeing where they go would be good. I like the idea of looking at the damage of the leaf. You know, they have a preference of probably have eaten more of that leaf, but it might just be that they, you know, maybe, but did they go to it or do they just eat more of it? That's another question that kind of needs to be thought about. And so how do you monitor all that? You're just gonna have a, and then again, you talked about having a couple plants.
They grow more on infected plants.
We definitely know that, but do they have a preference to feeding on infected plants? Meaning, do they choose it? Like, am I going to choose pepperoni pizza over cheese pizza or cheese pizza with broccoli on it? That's my experiment. How are you going?
So basically what you're saying is that you tested out that the original hypothesis that it will grow bigger weight, more weight.
Yeah, we tested that. Yep, you're right.
So now we're trying to see what are different ways to test this hypothesis.
Right. So how do they choose? How do we know that they chose? Will they choose virus infected leaves more than non-infected leaves?
Right.
And how do we do that experiment? So that's a question I have for you all, is how would you design that experiment? Because maybe you will do a preference assay in this test. Maybe we'll look at different substances like nicotine and see if they have a preference for nicotine over non, you know, a Cuba diet without nicotine. How would you design that experiment? So those are the kind of questions that. And I like the idea, like if you did have a bunch of pizzas in this room, we could look and see if there was a preference for one type of pizza over another. Maybe pizza with anchovies versus mushrooms. There might be a different preference. You know, just depends a little bit. Or Domino's pizza versus Little Caesars. We can see who eats what pizza gets eaten the most. But you have to. But if you don't design the experiment right now, just have Domino's pizza on top and Little Caesars hidden at the bottom. There's probably going to be Domino's that's eaten first because nobody's noticed the Little Caesars present. So you have to really think about how you design these experiments. So that's what I posed to these students here. The way we did it for this experiment, and again, there's lots of different ways, is we collected leaf discs from virus infected plants and non-infected plants. And I do think it takes a lot more than two, right? You need to have a good replication. So the students are collecting the leaf discs. And in our case, we actually put them in a petri dish. And so each one of these discs had a different treatment. So in this area, we had virus, non-damaged. Remember, damaged means that I've went in and wounded the plant previously, punched holes in it, make it feel like it's beetle feeding on it. So we're going a little bit further than what that hypothesis was. Then we have virus damaged plants. That's this right here. Notice how it's a little bit, it's not as green. You see that? And then we have number 3, no virus. So this is non-infected, but it's been damaged. So it came from a plant that maybe had the deuce defenses stimulated. And then we have number 4, which is no virus, no damage. So now the beetle has to run around in this petri dish, the simulated arena, and we'll see which of these leaf discs it prefers to eat. Now, waiting for a beetle to do its thing can be pretty laborious. So we actually ended up doing what you suggested, which is we measured the amount of damage caused on the leaf. So this is the amount of damage on leaves. This student, and again, if we had the right software at this time, maybe that would have been a better way to do it, or maybe weigh the leaf disc. That might have been a better way to do it. Because a whole leaf disc is going to weigh differently than a one that's been eaten in half, right? So that would have been maybe a good way to do it, like you kind of maybe hinted at, or just examine the leaves for the amount of damage. Now, this student was blind to this study, so she doesn't know what she's looking at other than she's trying to determine the percentage of damage on feeding damage on the disc. But she doesn't know if she's looking at virus infected plants or non-infected leaves. She just looks at one and writes down how many leaf discs were damaged and not, 2, and so forth. So these are the discs that she looked at. And so the easier way to look at it is to look at the leaves that are intact completely versus the ones that are damaged. So here's one that's clearly damaged. You'll see that. So we got like 1, 2, 3, 4, 5 that aren't damaged. We have like maybe 2 that aren't damaged here. But the no virus, we have 1, 2, 3, 4, 5, 6, 7, 8, a lot that aren't damaged. You see the difference? And then the no virus, again, another no virus, no damage. Most of the leaf discs aren't really damaged, relatively speaking. Do you see kind of, I mean, I'm not trying to make you drink the Kool-Aid. Do you see a difference between these at all? Again, they still definitely eat no virus as well, but there's a lot more discs that weren't touched. So they spent more time eating virus infected leaf discs than non-infected, but they'll still run around. So that means that, and so what it does suggest is that there is a preference for virus infected plants. It's statistically different based on the way we did it. We're looking at probably about, we got about 20% damage. So we're looking at probably about 30 or 40% more damage for the virus infected leaf disc. It didn't seem like it didn't seem like the previous damage made any difference. So we just basically grouped these all together. And again, this is just for bean pod monovirus, but anyway, we saw the same result for southern bean mosaic virus as well. Again, this is the number of replicates, 40. This is the percentage of damage, the mean, and then this is the standard error. And so they're not overlapping. We've done a statistical test, which is what we call an ANOVA, followed by a Fisher's least significant difference. And we found that the probability was less than 5. So the question was, what was making the leaves more attractive? So I asked another question is like, is it the color of the leaf? Is it the smell of the leaf? And so we put the leaves in dark and they seem like they don't feed very well in the dark. So I can't really rule out how much lighting is taking place. So there seems like that preference that we saw doesn't exist in the dark. The preference seems to be light-based, the visual cues of the leaf disc. It seems like that anyway. But this made us start to think a little bit, were they just less active in the dark? Are beetles less active in the dark? How would you test that? How would you develop an experiment to test whether or not beetles are active in the dark or can't find leaves in the dark? Well, one of the problems with being in the dark is you can't see what's going on. Real darkness is you can't see anything. And I don't know about infrared cameras and all that kind of stuff wasn't available to me. I couldn't go to Shields and buy a monoscope and look at their temperatures and watch them or anything like that. There's no infrared cameras at this time anyway. So my point though is how would you scientists, how would you discover or measure their activity in the dark? Yeah.
You would basically have a beetle, kind of like the petri dish, I guess. You can kind of have a beetle with a leaf and a petri dish and light, and then test the same in the dark and monitor the activity also by the. I guess can you monitor the activity by eating as well?
Yeah, that would be one thing. Eating activity is different though in walking activity, right?
Okay.
Yeah, so you have a point. So you would measure you can measure light versus dark. And so we kind of did that before. And this, what we're seeing here is they ate a lot less in the dark than in this experiment, which was in the light. But I was just curious, like, is it just they didn't walk around and find the leaf? Maybe they were just so clueless where to go in the dark that they just sat there. So I was just curious if they were walking around. And how would you measure whether or not they're walking around? Can you think of an easy way to do that? Again, we don't have any specialized equipment.
If there's no lights, like damage to the leaves.
Yeah, well, let's just say that we don't even have the leaves in there. How do we know if they're walking around? Yeah.
Is there anything we can just put in there to show the demarcation, to show that they move from one place to another? Is there anything we can add
To the beetle, maybe? Yeah. What do you think you can add to it? Paint its feet, maybe? Well, these were all things that we're trying, but the problem is the camera is that when it's whole darkness, you can't see anything. And I don't know about infrared cameras and all that stuff that wasn't available.
Some stains.
What's that?
Maybe stains or
Yeah.
stain the beetle and wherever it was in this.
Yeah. So these were all ideas that we had too. The other thing we thought about was whether or not if we just left like a little sand on the ground or you remember the carborundum stuff that's like really that light gray stuff. But the problem is they started choking on that and dying when they had the carborundum in there, just filled up their spiracles or something. And if you paint their feet, the paint dries so quickly. And then we thought, well, maybe we'll play with the auger and make it really soft and maybe we can see little beetle footprints. But that didn't work very well either. But the funny thing is anybody ever made an auger plate? Sometimes they can leave a little film of water moisture on the top. And we found that moisture stayed there and that if they walked on it, they would leave trails. So we accidentally figured out a way to measure their ability to walk in the dark. We basically made a fine mist of water on the petri dish And then we can watch their trails. So that's how we figured out whether or not they're active in the dark, see the trails of the beetle. So anyway, what the point is that they definitely move around in the dark. And as you mentioned before, we even put them on a cup with just a leaf right on it. So they're really sitting right on the leaf. And they will eat in the dark as well. But they seem to have trouble finding a leaf disc in the dark. Or any particular leaf disc. Does that make sense? But the point is these are the ways that we solve some of our problems. It's just a lot of times there's a little bit of serendipity and troubleshooting, because there's a lot of troubleshooting in science. There was no straight line answer. This is how we're going to do it and it's going to work. Just like with me doing the surgeries, I killed a lot of caterpillars before I figured out how to do the surgery. Luckily, I didn't have to go through any review boards or anything like you would with animals with backbones. All right, so you guys ready for a little break? Let's take a little break and then we'll try to go to at least 5:30-ish. Sound good? All right, so the next side of all this is what's happening to the, now we looked and saw that the plants grow better, or excuse me, the beetles grow better when they feed on virus infected plants. We saw that they had a preference. Now the next question is, does a beetle fed on plant more likely to be resistant to infection or more susceptible to infection? So the beetle's chewing away, eating all the leaf. Does that plant have a stronger antiviral response or weaker resistance to the virus? And then we go back to this slide here, and we can say, well, if the plant is being fed on by the beetle, is going to turn on all these defenses and suppress these defenses, making the plant more susceptible. That's the hypothesis I would sell to you. But we got to do the experiment and see if I'm correct. Regardless if I'm correct or not, as long as I have some difference, then I can write up a story. Okay. So what we did was so again, our objective is we are interested in determining whether a virus and its beetle vector benefit each other by co-suppressing the plant resistance towards the virus. So we're going to determine if bean plants fed on by the Mexican bean beetle are more susceptible to bean pod monovirus or southern bean mosaic virus. Again, that's my hypothesis. That's what I think is going to happen. And so what we did for this research was that we used clip cages to hold one leaf, and we had let the beetle feed on that leaf, or we left it non-wounded, or we wounded it with a garlic press where we punched a bunch of holes into it to simulate herbivory. Remember how I like to punch holes in the leaves? We pretend to be a caterpillar. Well, I pretend to be a beetle now too. And so that's what we're doing. So these little medicine caps are now our clip cages. We got some foam on them so we don't damage the leaf. We're using younger plants because they're more likely to become infected. Here we are collecting plant sap. So after we've done that setup, we've wounded plants with beetles, we've wounded plants with garlic press, or we left them alone. We're now going to put some garlic sap on, or excuse me, we're going to use some plant sap that's from infected plants at a very low amount, and then we're just going to rub the leaf and see if the plant becomes infected after these treatments. And again, we used a very small amount of sap, because if you use a ton of sap, you're going to infect it no matter what. So we used just enough sap to get about 50% infection rate. We used a garlic press to smash the leaves that had virus infection, and then we diluted it with water. And then we painted the leaf disc. Here's the carborundum, that's that gray stuff. We gently rubbed the leaf with the virus. And what we found was beetle wounding actually reduced the percentage of infected plants. And artificial wounding, smashing the leaves, reduced the amount of infected plants, not as much as beetle feeding, though. Beetle feeding really reduced it. While non-wounded plants were the ones that were most likely to be infected. In fact, they're about twice as much or more than twice as much infection rate. This is the percentage of plants. This is the mean. This shows you a significant difference. These letters mean significant difference. So what did you find out? What was my hypothesis again? My hypothesis was that the beetle plants, the plants that were previously fed on by beetles should be infected more. What did I find out? The plants fed on by beetles were actually more resistant to the virus, opposite of what I was hoping for, in quotes. Remember, I'm objective, I'm a scientist. So the alternative hypothesis came out. Does that make sense to everybody? So here's an example where I had an idea and reality was different. The reality was that beetle feeding or artificial wounding that kind of simulates beetle feeding. Remember, there's something in their spit that might be doing something different. And the damage is a little bit different. But the wounding and the beetle feeding make the plants more resistant to virus infection. Not more susceptible, more resistant. Opposite of what I thought was going to happen. So my hypothesis was that they were going to be more susceptible. The null hypothesis, there'd be no difference. The alternative hypothesis was demonstrated to be correct in this experiment. So that's no problem. I'll just write a paper that says this is what happens. Yeah.
Does it make the plants more prone to the same type of virus or different?
Well, the only viruses we tested was, I think in this case, bean pod model virus or southern bean mosaic virus. I'd have to go back and see. But it made, I don't know about other viruses though. And generally speaking, we would think that a wounded plant by caterpillar, for instance, would be more susceptible to some other type of viral infection. That's what we would normally think because of the crosstalk. But in this case, the wounding apparently made the leaves more resistant to the virus. Not what I was expecting. So then I just had to come up with an explanation on why that's the case. But apparently the virus infected plants do help beetles to grow better or faster. But the beetles aren't necessarily making the plants more susceptible to virus. Is it a mutualistic relationship if the beetle transmits a virus and allows them to grow better? Maybe it's kind of mutualistic. So here's the kicker. In beetle regurgitant, they have an enzyme called ribonuclease. Remember how I was interested in spit? Well, in caterpillars, they have glucose oxidase. In this beetle, one of the primary defenses is ribonuclease. And what does ribonuclease do? Does anybody know what an RNase does? What does an RNase do? Or ribonuclease, what does it do? Yeah.
Destroys RNA.
Exactly. So this virus or this beetle is spreading a enzyme in their regurgitant that destroys RNA. And believe it or not, these are RNA viruses. So that's pretty crazy. But these particular viruses seem to be able to overcome that regurgitant. When I say regurgitant, that means I'm squeezing the beetle. They don't have little salivary glands I can dissect out. And so I'm collecting the sap So I'm collecting the beetle regurgitant. So anyway, here's the garlic press. This student's punching holes in the leaves. And she's going to paint on ribonuclease. Or she's going to paint on buffer, just water and some salts in it. The same stuff that the ribonuclease is in. Or we're going to let beetles to feed and caterpillar feeding to take place. So what are we testing here? We're trying to get at whether or not this ribonuclease that's present in beetle spit may have an effect that goes further than the wounding. Does that make sense? So the garlic press is punching holes into the leaf to simulate beetle feeding. In some cases, we're going to paint on ribonuclease. In other cases, we're just going to paint on water. We're going to see if ribonuclease does something to these plant defenses. We're also going to have some beetles feeding, and for, I guess, shits and grins, we're going to have the caterpillar feed also. And so here's what we found out. This is looking at the number of lesions on a leaf, so it kind of gives us an idea of infection rate. The non-wounded plant had the greatest number of lesions. So again, non-wounded plants are more susceptible to the virus than beetle feeding. Does that make sense? So beetle feeding makes the plants more resistant to the virus. Artificially wounding the leaf and painting on water doesn't seem to really make the plants that much more resistant to the virus. They're still pretty susceptible. But if you paint on ribonuclease, look how much more resistant the plant becomes to the virus in comparison. So here's the non-wounded plant. Here's artificially wounded, treated with water, artificially wounded and treated with the ribonuclease. A lot less lesions, so a lot less infection. So it may be that the beetle's regurgitant does have an effect, because you see the levels are equal to the beetle wounding this time. And surprisingly or interestingly enough, caterpillar wounding also makes the plants more resistant to the virus. So whatever happens when this plant gets wounded, particularly if there's spit involved from either the caterpillar or the beetle or ribonuclease, it seems to make the plant more resistant to the virus. Believe it or not, all these experiments are actually can be kind of a pain in the behind doing all this. And you still don't have a completely satisfying answer. It feels like it never ends, basically. Anyway, but I think it was interesting. It was a really cool experiment. It was fun to include students. It was fun to get a publication out of this. It did seem to have some notice because of the fact that ribonuclease was now shown as a potential chemical, insect-derived chemical that may affect plant defenses. That's what made it kind of interesting. But here's an example where an alternative hypothesis came through and it still provided evidence enough to publish. And it was an interesting publication. The main thing is you got to have strong methods and strong enough results to publish. Doesn't have to be the perfect result, but you have to have something that's clearly followable to make it into, and this will get into a decent little journal. Big journals would probably require me to start measuring the proteins and the genes behind all this and yada yada, and it'd go on and to get it into something really big. So anyway, bean leaves treated with ribonuclease and fed upon by these insects had increased virus resistance. It wasn't what I suspected, but that's what happened. And so that I was publishing what happens and then try to explain it. And I could probably put it into an evolutionary story. The evolutionary story I would have is that plants, when they get fed on by insects, are more susceptible to being infected. So it makes sense to turn on proteins that help protect you from infection. So when you cut yourself, you're going to turn on immune system and bring in immune cells that help prevent an infection in that wound, right? So same thing with the plant. When the plant was wounded, it obviously turned on some proteins and defenses that help protect it from being infected. If a beetle's coming along, it's a good signal to the plant that I'm more likely to become virus infected. So I better turn on some defenses. Now, how come you don't just turn on your defenses all the time in a plant? Well, the problem of turning on your defenses all the time is if you're always in a fight mode, you're not growing as well. That's why you have induced defenses. It's not there all the time because you're wasting energy, precious energy that you could be spending on growing and ultimately making seeds for the next generation. Because what do we know about evolution? It's fitness that matters. Fitness meaning how many progeny you leave. Now, some plants live in such a harsh environment, they got to have plant defenses all the time because they grow so slowly anyway. Desert plants tend to be very focused on making defenses like thorns and spines and more toxic because their growing season is really, you know, they can't just grow willy-nilly. They got to wait for rain, and if they get eaten, and they don't grow very fast, so they're going to have to really protect themselves as smaller plants until they get bigger. And even then, they focus a lot more on defenses. So my point is, there's all sorts of evolutionary strategies for different types of plants. Some plants aren't particularly nutritious. Grasses, for instance, aren't particularly nutritious. So they grow fast. Some plants, there are strategies to grow really fast and spend less energy on toxins. Some plants like nightshades and tobacco, they spend a lot more energy on defenses, chemical defenses, because they're more nutritious as a leaf. You ever looked at a grass blade versus a tomato leaf? There's a big difference, isn't there? So a tomato leaf is really juicy. So they spend a lot, that plant spends a lot more energy making its defenses than grass. Interestingly enough, grass, if you have some sheep or whatever feed on it, there's enough spit in the sheep spit when it's eating, it'll actually make the grass grow faster than if you just cut it with a mower or something like that. So plants definitely respond and they have different strategies. So you can make, so I make the evolutionary argument that when a plant is being wounded, it's a signal, particularly if it's from a beetle, that I better be ready to deal with a virus infection. So I'm going to go ahead and start stimulating my viral resistance. I'm not going to wait because then it's too late, right? So anyway, as I say, that's that. Anybody have any questions? Does anybody wish we'd have a really serious winter? At least one serious snow day? All right.